All right, guys, this should finish up the end of your taste and smell notes. So we're going to talk about taste here for a minute, or gustation. So find that section in your notes, and here we go. We start with the anatomy. What are the parts that are involved in taste? Well, we think about our tongue, we think about taste buds. So all of these little tiny dots right here would be taste buds that you think of and see. You actually have three different kinds of taste buds. So the ones that are on your tongue right here, these are called papillae. And specifically, they are called fungiform papillae. So these cover the majority of the surface of the tongue, and fungiform means they look like a little mushroom. So if you were to zoom in on this, it has a little mushroom-like bump shape. Think about like a mushroom with the cap on it that you would buy at the grocery store. Each one of these little bumps kind of resembles that. So a fungiform papillae are definitely the most common type of taste buds that we have, and the ones that you probably think of. Now, you also have these foliate papillae back here on the sides of your tongue. If you can see where my cursor is, they almost look like little tiny slits. So they're at the very back of the tongue before we hit kind of all of these bumps at the way back of our tongue. The last type of papillae would be a circumvallate, and there's a small row of these at the very back of your tongue that sort of divides what we think of as the majority of our tongue with these giant bumps back here, which I'm going to talk about in just a second. So you have one row, they're kind of like an upside down V shaped, and they sit right in front of the big bumps in the back. If you look at this diagram and we sort of grab one of these and blow it up, here is that bump that we see. And if you zoom into the side of the bump, we get this picture. There's a cartoon version, at least, of this picture. Down here is a real life version. So within one of these little sections, we can see this cluster of cells, and that's actually what we would call a taste bud. So when we use the words taste bud right now, we probably are not using those accurately. We use them to represent these fungiform papillae, these bumps on our tongue. In reality, they are these sort of cluster of cells that are embedded within the papillae on our tongue. And here is the circumvallate to use as an example. So 3 different kinds of taste buds. You need to know their names and you need to know their location. Now let's talk about the actual taste bud here and a couple of important structures. So let's start with sort of follow if a chemical were dissolved into saliva, what would happen? It would enter this little hole in the overall taste bud, and that's called a taste pore. So that gains access to chemicals dissolved in saliva would gain access into this little taste bud by way of the taste pore. Inside, every single one of these little purple cells with the nucleus, that is a gustatory receptor cell. So here are this is the connection to the nervous system. These are chemoreceptors, and they have little tiny cilia or gustatory hairs, just like we saw in the nose, on the ends. So a gustatory receptor cell are the purple cells within the taste bud. If we jump back to sort of the tongue area, let's talk about tonsils for a second. So we think about tonsils, we actually think about these tonsils that are here on the sides of our throat. That is one type of tonsil, but we also have tonsils at the very, 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 very back of our tongue that are hard to see unless you stick your tongue out way far. So the ones that are on the sides of our throat are called palatine tonsils. When we look in the back, if we see little swollen lumps kind of at the back of our throat, those are palatine tonsils. These are what typically can be removed as a kid. If you've ever had your tonsils and or adenoids removed, you had your palatine tonsils removed. The last is, let me collapse this, set of tonsils are your lingual, and those are back here on your tongue. Another name lingual tonsils are filiform tonsils. So we could recognize both of those. If we Let's see, move forward. There we go. Here is just a large diagram to make sure that we have labeled all of the things that we need to label. So really quickly, I'm going to attempt to circle and or highlight areas that you need to make sure that you have labeled on your diagram. So we need our papillae, right? Foliate, fungiform, and circumvallate, which, yes, this picture shows a blown up version, but make sure you connect it back to that image. We need our 2 sets of tonsils, so make sure you grab your tonsils and you label those on your diagram. We need over here, within the taste bud, we need to know what is the taste pore. We're going to see that again in a second. Those gustatory hairs and these purple receptor cells. Okay, those are the main things to label. This right here, the structure, the epiglottis, this is a really important structure that closes over your every time you swallow, you want food to go down in your digestive system. You want it to go down your esophagus into your stomach and hitch a ride through the digestive system. To make sure that it doesn't go down into your lungs, this little flap of tissue will close the top of your trachea so that you it's actually your nasopharynx, but it will actually close that tube system so that food doesn't go down into your lungs. So those are important things to make sure that you have on this particular diagram. All right, let's go see if I can figure out how to make this go back to a mouse. There we go. Okay, so, oh, gave us all of, oh no, look at this. It's going to make me erase. So let's go to that eraser and clear the whole screen. My goodness. All right. So let's talk about ideas for how we taste. Let me put a bunch of these up here on the screen. Okay, I need my picture. There we go. Okay, so 2 different kind of ideas on how we taste, or taste theories. They are competing ideas, and one sort of has replaced the other. So the first theory was called the regions of taste theory. It was developed in 1901. And this theory for how we taste and what we taste basically took the tongue and broke it up into regions and said, okay, the taste buds here on the tip of the tongue are detecting sweet chemicals dissolved in saliva. Those on the sides towards the front are sour, back sides are salty. The very back of our tongue detects bitter taste, and the center detects umami, which is sort of like a savory, think of like steak sauce and beef, a very savory chemical. So this theory said that there are different types of taste buds and they live in these certain pockets, and depending on what taste bud it is, it sort of detects different flavors. Now, we now know that that's no longer accurate. Instead, we have taste buds all over the tongue, and within every taste bud, we have structures that look like this. So here is without its boundary, here's the taste bud and the receptor cells that live in it. Notice the different colors, right? So this is saying that essentially every taste bud is capable of detecting every single taste flavor profile. So like there are receptor cells in here that can pick up sweet and sour and salty and bitter and umami all within one taste bud. And then these are everywhere all over the tongue. So essentially we can detect tastes, all these different tastes all over the tongue, not just in certain places. Okay, now let's talk physiology. This is basically the pathway that it takes chemicals being dissolved in a solution to sort of stimulate and travel up to our brain so that we know what we tasted. So it all starts again, because these are chemoreceptors. It starts with a chemical dissolved in saliva. That saliva then enters the taste pore, and the fluid, which has the chemical that you came out of the food or whatever you put in your mouth, is dissolved in the saliva, that's going to stimulate the gustatory hair receptors on the receptor cells. And you can see these cells make direct contact or synapse with a cranial nerve or a couple of cranial nerves. So then what cranial nerves are of interest in gustation? Well, the facial nerve is of interest because it supports the anterior, which remember is the front, 2/3 of the tongue. So any of the receptor cells in the taste buds up here, these are going to synapse with, make a connection to the facial nerve and carry that information up to your brain. The back third of the tongue is going to be innervated by the glossopharyngeal cranial nerve. And then the very back structures here, including the epiglottis, that's going to be controlled by the vagus nerve, the wandering nerve, cranial nerve number 10, that cruises beyond the head and the neck region. So depending on what's being stimulated as a part of the taste and swallowing pathway, we have cranial nerve stimulation. And then eventually those cranial nerves we saw when we learned about the brain, those are going to make their way up to the gustatory cortex, sensory cortex in the temporal lobe. So that's sort of where the pathway ends and where your brain begins to say, this is what I just tasted. It interprets the smell or the taste sensation. So that's the end of our notes. Taste and smell are very similar because they are both chemoreceptor pathways. Chemicals have to be dissolved in a solution in mucus in our nose and saliva in our mouth to actually stimulate the receptor cells. In both of those pathways, the receptor cells have little gustatory hairs on the end that sort of detect the chemicals in the solution